Hi everyone. Let's wait a few minutes to start, to wait for everyone to enter. Thank you. I'll start in about 2 minutes. Hi everyone. Just 1 more minute to see if we get the most participants on. Okay, I think we can start. We already have quite a few participants. We have around 84 people in the audience, so thank you very much for joining. Hello, everyone. So welcome for today's workshop on advancing remote sensing for macroalgae and seagrass ecosystems. This is organized under the EU-funded Bioecocean project. My name is André Valente from the Air Center, and I'll be hosting you through this session. So today we will explore the frontiers of remote sensing for macroalgae and seagrass ecosystems with a focus on the capabilities and challenge of the Copernicus Sentinel2 mission. As many of you know, the enhanced spatial resolution up to 10 meters of this mission and its open access data policy provides unprecedented opportunities to observe macroalgae and seagrass. Nevertheless, we still have important questions around spectral and spatial resolutions, validation, and even operationalization. So together, we'll be looking into the latest innovations, tools, and use cases that can help us make the most of this mission in monitoring macroalgae and seagrass. I am very happy because we have a distinguished panel of speakers, which will focus on very different environmental matrices, from submerged and intertidal habitats of macroalgae and seagrass to beach casts of macroalgae, and finally sea surface floating macroalgae. Before we start, I also would like to contribute by sharing your insights through an online survey. We are going to put the link in the chat now. So the survey will be open until next week. You can fill it after the workshop. We will be happy to have your opinion to help us in developing best practices and guidance for using Sentinel2 in monitoring macroalgae and seagrass. So thank you very much for helping us shape a shared vision going forward. So now, because we have a very tight schedule, let's dive right in with our round of presentations. So the program is, we have a round of 10 minutes presentations for each speaker. Then we have question and answers from the audience, about 15 minutes. And then finally, 30 minutes roundtable discussion with the speakers. So first in the agenda, we have Lina Nordlund from Uppsala University, who will present the Bioecocean project advancing biological and ecosystem ocean observations. Lina is the coordinator of the Bioeco-Ocean project. We are very happy to have her here. She will provide a quick introduction to the project and to this issue. She's a passionate and renowned researcher in seagrass and has an extensive record of publications in this field. Lina, please go ahead, share your presentation and go ahead. Thank you.
Thank you so much. Great introduction. Thank you, Andre, and thanks to Air Center for arranging this workshop. As Andre said, I'm the project coordinator for Bioeco-Ocean, and I'm also an associate professor in natural resources and sustainable development at Uppsala University in Sweden. And we have several partners in this project, and we started about just over a year ago. And there at the top, you can see our email, and you're welcome to contact us. And the ultimate goal of Bioeco-Ocean is to enhance biology and ecosystem ocean observing capacity for advancing scientific understanding of the ocean and increase the utility of ocean observations. And I think this fits really well with our theme and topic today. So how do we meet these ultimate goals? We think in Bioeco-Ocean that what we really need is coordination and interoperability. But how do we do this? And we think and suggest that more consistent communication and collaboration among sectors and stakeholders will take us at least one step further towards these goals. We have a co-creative process. It means that from the very beginning, we are developing a blueprint together with you, and we are receiving feedback. We are furthering developing this blueprint. And the blueprint is a question-based support tool. So, as I said, this is a co-creative process. This is under development. So the question-based support tool, it's there to support our thinking, and it's not a prescription on what to do. So it doesn't going to help us to tell us exactly what to do. It will help us to spark and encourage collaboration through these questions and facilitate communication across sectors and stakeholders. And ideally, it will also enable future visions on how we can do more integrated approaches and collaborations. So the blueprint will be launched on a digital platform. It will be interactive. There will be training materials, and it's planned to be launched in September 2027. And the blueprint, as I said, it's evolving through this co-creative process, and we don't know exactly where it's going to end up, and I think that is also very exciting. You are all invited to be part of this co-creative process, and I will tell you a little bit more about the blueprint in a little while. But first, I want to say a few words about essential ocean variables. So the Global Ocean Observing System at IOC UNESCO, they have a couple of or 3 panels with different essential ocean variables. And the idea behind the essential ocean variables is that they can describe a lot about the ocean. They're collecting primary data. As a foundation in the Bioeco-Ocean project, we have the Biology and Ecosystem EOVs. And together with GOOS, the Global Ocean Observing System, we have updated the specification sheets for these EOVs. So it's basically the recipe of how to use them and what to do. Of course, seagrass is one of the essential ocean variables, as well as macroalgae. And we have the macroalgae cover and composition and the seagrass cover and composition. And they consist of subvariables. And both of these essential ocean variables has areal extent, which is very relevant for the work that we are discussing today. Another subvariable is the percent cover. And imagine if we have these essential ocean variables, these things are being measured the same way across the world, and they're making they're becoming openly available. We have a lot of ground truthing data out there. So the dream, have a lot of sustained monitoring, all observations becoming available globally through a shared data ecosystem that I know Elizabeth will tell us a little bit more about soon. And then the sort of the amazingness of having this global digital ecosystem, that the data would be available, that we can make more and better products. Coming back to the blueprint, so how is that related to the essential ocean variables and to the work that we are discussing today? So the blueprint consists of a lot of different components. And if we look, for example, at the application and society and data products, these two are very closely linked, right? Are we creating the right products? Is the products actually meeting the needs of researchers, of managers, of policymakers? Do the data products have enough accuracy or resolution for being usable? So these are the question-driven things that will appear in the blueprint. Another important link is the data products and the data collection. Are we actually collecting data in the right places to create the best products? Are we collecting the right data also to actually create the products that we want to do that are relevant for society, for example? And with the blueprint, the more components that we are putting together, the more complicated it becomes. But answering these questions, working together across stakeholders, across the sectors, can really help us advance our understanding of the ocean and marine life. Lastly, I just want to bring this picture back that with a shared digital ecosystem where data is made fair, we will have a much better opportunity to create amazing products that can be used globally. So that was just a short introduction to the BioEco-Ocean project. Thank you.
Yes, Lina, thank you so much. It was a perfect introduction to BioEco-Ocean project and our objectives. So now moving on to our next speaker, we have Elizabeth Lawrence from OBIS and UNESCO, who will present powering remote sensing with OBIS data. Elizabeth is part of the technical team in OBIS, the Ocean Biodiversity Information System, which she will probably explain a bit more soon. She is very well rounded in the complex issues of managing biological and biodiversity data. So Elizabeth, the floor is yours. Please go ahead.
All right, thank you, Andre, for that introduction. Yeah, so today I'm going to be talking a little bit about what kind of data is in OBIS, specifically what kind of macroalgae data is available to be used. So first, to give a little bit of background on what is OBIS. So OBIS is the Ocean Biodiversity Information System. It's the world's largest open access repository for standardized marine biodiversity data. And I really like to emphasize the standardized part of this, because even though there's over 5,000 different data sets, all of these data sets are integrated and standardized. And so they have the same data columns, the same data headers. So it's very easy to use any number of these over 167 million observation records. But there is more than just species occurrences in OBIS. So there's over 181 million different types of measurements available in OBIS, and I'll show some of that a little bit later. There are measurements related to DNA-derived observations, so like eDNA of like macroalgae, seagrass. You're going to have habitat data, information on ecosystems, abundance, biomass, satellite-derived or tracking data, as well as measurements derived from imaging. And so, of course, OBIS has a lot of different types of data. And so we're going to take a look at in situ data, some polygon data, image-derived, and measurement data in OBIS. Starting with in situ. So, of course, the in situ data in OBIS can be used to help validate remote sensing. So minimally, all data in OBIS includes information on the taxonomy, the location, so the coordinates, depth, and as well as the date the taxon was observed. And so here I'm showing a snapshot of all macroalgae and seagrass data in OBIS. And this data comes between 1648 and 2024. So we have a lot of historical records as well. And this map is almost 200 million presence records of macroalgae and seagrass. Not shown in this map, but also available in OBIS. There's almost 2.7 million absence records. So there are presence and absence records available in OBIS. And this map is represented by 2,300 different taxa. So these are whether the organisms are identified down to the species level or genus, family, and so on. And then just for fun, I've included this table here showing the top 3 macroalgae families represented in OBIS. So moving on to the polygon data. So although OBIS can't hold like the shapefile data itself, so there's no way to include shapefiles or KML files. It is possible to include well-known text strings. So these are similar to KML files, and I believe they can be converted somewhat easily. But they're just strings that can define lines, points, polygons, or a collection of any number of these. And then in OBIS, this data is found in the column footprint WKT. And so this map here on the right, I'm showing just over 25,000 valid WKT strings. And so I'm only showing lines, polygons, and geom collections. So I've excluded any WKT strings that were just points. So you kind of get a little bit, and I've zoomed in here on Europe, so you can see what is available, or you can get an idea of what kind of polygon information is available when it's provided. As for image data in OBIS, unfortunately, we cannot hold the images themselves currently, although it is something we're exploring. But OBIS can hold image-derived data. And the links to any of the raw images are available when data providers provide them. So these links are found in the associated media column. And this image here is an example from a data set in OBIS of what kind of links can be included. And this also is a map. So I did some filtering on that previous heat map that we saw. And These are all of the data sets and all the occurrences of macroalgae and seagrass and obis that have included something in the associated media column. So it's only coming from 10 macroalgae data sets and just over 5,000 occurrences, which may not be a lot compared to the number of data. So sometimes you can find this information in the data set metadata rather than this column itself. So I mentioned in the beginning that there's a lot of measurement data in OBIS, which can be really useful for looking at abundance or biomass. So here we're looking at the genus Zostera, and I was able to obtain over 62,000 different measurements for this genus. And I've listed the top 5 measurements here, as well as how many observations for each. So the most common measurement is the greatest abundance within a transect quadrat sample, which actually had to do with the abundance of the like the sediment type when I went to actually look at what that data looked like. And here, so what I've done is I'm showing the sampling device. Although sampling device wasn't one of the top 5 measurements, it was a pretty common one. And so what I've done is I'm showing this map of all of the data sets that have used this link here as an identifier specifically for the sampling instrument name. And so you can see there's a variety of different sampling devices used to collect Zostera information in OBIS. Varies from cameras, observers, different types of cores, quadrats, and videos. So that can be pretty interesting. So there are some limitations in some OBIS data. So I think one of the most obvious is just first any gaps in data coverage. We only have data on what has been published to OBIS. So we're always happy to help people fill in gaps. Also, sometimes data providers don't always include WKT strings or the associated media. So sometimes, again, that might be in the the data set to metadata, and you might have to look elsewhere for that information. And another limitation would be the inconsistent use of controlled vocabulary, which can help facilitate data set filtering. So if not everyone is using the same names, it can make it slightly tricky. And I just wanted to mention some future work we're working on in OBIS is adding some dataset tags. So it's really easy to find datasets that are fit for particular use. We're working on improving filtering options, make it easier, and in general, improving the EMOF, which is the table that holds all those measurements, and making it easier and more manageable to deal with and get information from. So that is my presentation. I'll be happy to answer questions later.
Thank you, Elizabeth. It was, I think, very clear, the power of OBIS and the world that it's OBIS. I think it's a very useful resource that needs exploration. So now it's my turn. As I said, my name is André Valente, and I'm from the Air Center. And I will present mapping intertidal macroalgae with Sentinel2 in BioEcocean's Atlantic Living Lab. So I'm also involved in the BioEcocean project in the part of remote sensing. My background is in physical oceanography, but for most of my research has been focused on using satellite data to understand the physical biological interactions in the coastal and open ocean. I'm going here to share my presentation. Okay, so this is a study we are doing in BioEcocean. It's a collaboration with CIMAR and their center. There are several people involved. Just going to give you here a first introduction that most of you already know. So macroalgal forests are key habitats providing vital service for ocean ecosystems and humanity, from carbon sequestration, food supply, habitat provision, nutrient cycling, and many, many other services. So because of its importance and impact, and because it's feasible to observe, macroalgae cover and composition has been recognized as an essential ocean variable to monitor, among many others. But here I'm going to follow this one. So in Bioeco Ocean, there are several living labs. One of them is the Atlantic Living Lab, which objective is to advance research around ocean observations of macroalgal forests in the Atlantic Ocean. And the main focus is in the coast of Portugal. So this is a biogeographic limit for many species of macroalgae, with their southern and northernmost distribution limits in this area. And there are several studies ongoing on this lab, from environmental DNA, remote sensing, and passive bioacoustics. I'm going here to focus on the remote sensing study. So we are doing this study on the rocky shore of northern Portugal. This is Viana do Castelo. There is here a large intertidal area with also very wide macroalgae seaweed beds that are exposed at low tide. There are extensive beds of fuscus, and the southernmost population of Ascophilium nodosum in Europe. And some fundamental questions we want to answer that are not only for the remote sensing part, but generally is what are the macroalgae trends in this region? How are the trajectories different from other regions? What are the drivers of these changes? So how can we answer these questions? So field data and drone data can provide very detailed maps on macroalgae and seaweed coverage. However, they typically cover smaller areas, and they also require some human effort for monitoring over time. By contrast, this new recent mission from Copernicus since 2016, it offers an unprecedented open source, high resolution up to 10 meters satellite imagery, which opens possibilities for the long-term monitoring in these narrow and complex intertidal zones. So this is just a brief recap of Sentinel2. It's a constellation, mostly has been with 2 satellites, 2A and 2B. The 2C, a third satellite, has been recently launched last September, but it's still not operational, I believe. They are all similar sensors aboard, and MSC gives reflectance data at 13 bands, from the visual to short band, at different spatial resolutions. Some of them are 10 meters, and the lowest resolution is 60 meters. So it was a mission mostly designed for land use, but also has been increasingly applied in the coastal ocean. So this satellite is not giving data currently for the open ocean, only coastal areas, but you get the coastal global coverage more or less every 5 days. So with this new mission, we can potentially improve our capability to monitor seagrass and macroalgae over long periods of time and large areas. So here we combine this Sentinel2 imagery with field data to provide estimates of intertidal seaweed over large areas and long periods of time. How is the methodology, the recipe? So to start, we start with field data with the UV acquisitions and also quadrats in situ precise geofencing of independent training and validation areas. So this is in the bottom left, you see the high tide and the low tide. So on this day, we acquired both UAV data and quadrats. And from this data, you can develop maps of seaweed coverage. So it is based on a supervised classification technique from a previous work. A map of intertidal seaweed assemblage is created. It was originally with 5 classes, 3 different species of seaweed, 1 rock and 1 sand. There was a good classification level of agreement. And then because it's challenging from Sentinel to differentiate between the species, we merge the 3 species, so you get a combined binary classification map of seaweed and non-seaweed, which is your right map that you have there. So once you have that, how can we use this to apply and to develop models in Sentinel2? The first step is to resample the UV map to Sentinel2 resolution. So this is not that straightforward because the grid of Sentinel2, it's not regular, so not all the line of pixels or columns are the same lat longs. So every bounding pixel has different lat longs, but you do this registration of the pixels in each Sentinel2 grid pixel at 10 meters resolution. It gets you about 100,000 drawn pixels for each Sentinel pixel. And then what you do is you calculate the percentage of seaweed pixels in each Sentinel2 pixel. So you get a map of seaweed coverage percentage in the Sentinel2 resolution. This is your right map in the right side. So when you have 100% coverage, it means 100% of that pixel is covered by macroalgae and seaweed. So you get an area of 100 square meters. The next step is how to predict coverage based on Sentinel data. So for this, there are multiple methods here. We explore spectral index. So first you acquire a coincident Sentinel image. So this was also low tide. It was about 1 hour after the UV acquisition. So almost the same tide. And we use the operational product of level 2 data, and we calculate different spectral index. And then we try to relate this Sentinel2 data spectral index with the percentage coverage taken from the UV at the Sentinel pixel grid. So different models were tested and different index were tested, and our best approach was a polynomial relationship between the UAV-derived seaweed coverage and the Sentinel2 NDVI. There is a convincing relationship, and have a R-square of 0.83, and then for the validation we use 50%. So the curve is done with 50% of the data. The validation is done with the other 50%. It gives you a good mean square error of around 13% in coverage, which is not bad. So it gives us some convincing arguments to proceed forward. And then what was our next step was to apply over a large area. So the training area is this polygon here in the right side that you see in yellow. And then we apply the model to the NDVI of all this area in red, which is a mask manually made for the intertidal zone. You get the Sentinel2 pixel seaweed coverage on the right side. And we also overlap this with a survey done a few years before. Actually, this date I'm seeing here, it's a typo. It's not 2024 of April, it's 2021. And so for further validation, we used a field survey of the Ascophilium nodosum beds. These are the red contours that you see, and there is a good overlap between the areas with higher coverage of seaweed and these field surveys. The last and desired step is then to acquire long-term variability. So for this, we obtain 64 Sentinel image during fall to avoid green algae coverage, which grows at some periods of the year and may add uncertainties to this model. And all the 64 image were in the same optical atmospheric conditions, quite cloud-free and haze-free. And then we acquire the image, we apply the spectral index, and then we apply the model. Then what you get is in that training area, you sum all the area in the pixels. This is a very preliminary result, but it suggests some kind of decreasing changes in the seaweed coverage. But this is something that deserves more attention, and we will continue on this study. So as a summary, we improved the mapping of macroalgae coverage by combining UAV data in situ, geofreezing quadrats, and Sentinel2 imagery. Macroalgae coverage extent percentage in each Sentinel2 pixel is estimated based on a regression model between NDVI and the seaweed extent from UAV imagery. Model validation performed on a subset of the labeled data and a 2016 survey. This methodology was tested on assemblage dominated by Scophilium nodosum and Fuscus spp at northern Portugal. And potentially this methodology can be applied to monitoring spatial temperature changes in other regions and also assess the Atlantic extended carbon stocks and the effectiveness of seaweed restoration actions. We have lots of work ahead, including increasing training data with additional flights of drones and in situ measurements, testing other models. We also want to assess the impact of the green algae and aim to differentiate between macroalgae species and also move a bit forward to assess carbon and biomass estimations. Also, there are other methodologies, as you will see in next presentations. And finally, ideally, we want to produce an automated workflow that can help stakeholders. And that's it on my side. Thank you. I'm going to stop sharing. And we can now move forward to now. So now it's time to Dimitris Porcedanis from Forth, who will present Copernicus and what it can offer in the mapping and monitoring of subtidal seagrass habitats. Dimitris is an active and passionate scientist. He's been involved in seagrass research for many years and has many impactful papers on this issue. So we are very happy to have you here. Dimitris, please go ahead.
Hello, good morning, good afternoon. Nice to meet you. I will move the presentation about the subtidal seagrass mostly and what we have done so far with Copernicus Sentinel and other Copernicus contributed missions, which is quite important to add it here in the discussion. So I will share my presentation here now. Here it is. Okay. You see it?
Yes.
Great, thanks.
Perfect.
The topic for the next 10 minutes will be about the use of Copernicus Sentinel2 and Copernicus contributed missions in support of global seagrass mapping. How the previous speaker has talked already about Copernicus Sentinel2. Usually it's 2 polar orbiting satellites. Now we have 3 for 1 year. We will have every 2 to 3 days new imagery, so it's very good that we can aim close to hypertemporal data when we don't have clouds, of course. The sensors have been designed to monitor changes in land surface conditions. That means that are not optimized for water applications, but we take the advantage that they pass over our coastal zone, and luckily we can make use of that for our benefit. Copernicus contributed missions is something interesting that people are not aware about that. In addition to the data provided by the Sentinel satellites, the contributed missions play a crucial role in delivering complementary data. These are usually missions from ESA, from the member states, from EUMETSAT, and other European and international third-party missions operators that make some of their data available for Copernicus. We have around 30 existing or planned contributing missions that fall into the following categories, like the SAR data, optical sensors, altimetry systems, radiometers for land and ocean temperature monitoring, and spectrometers for measurements of air quality. Here we have a lot of commercial data that usually we have to pay for them, but through easier processes, you can write a small proposal and have access to high quality commercial data to supplement your projects. We have worked with Copernicus Sentinel2 in mapping country scale seagrass. Back to 2018, we have managed to map the Greek seagrass meadows using Copernicus Sentinel2, and at that time, Google Earth Engine platform. We make a quite substantial effort to make a harmonized product with some success. Of course, we learn a lot from that process, and we'll move forward to the next steps to the Mediterranean. This was a big challenge because computationally intensive. We spent almost 2 years by creating data, running, submitting proposals to Google to give us more computing power, and in the end up, we end to have the capability to map the meadows down to 20 meters depth because of a lot of complex processes that follow in this analysis. And we estimate an areal extent of seagrass meadows in the Mediterranean, mostly Posidonia, to a lesser extent Cappadocia, almost 19,000 square kilometers. Of course, this is not complete, given that we have this depth zone that we work, and there is a need for revision of such approaches when you move from local to country and regional scale, trying to do subdata seagrass mapping. After that, and in meanwhile, we move to East Africa, trying to support activities from WWF Germany in Mozambique in Quirimbas National Park. We apply similar tools using Google Earth Engine and Copernicus Sentinel2 temporal imagery. In this marine park, we use also bathymetry from space applications to estimate slopes and rugosity and bathyc position index to inform the coral reef side of the process here. And for the seagrass list, we make spectral mixing and hard classification to quantify at the pixel level sort of fraction cover analysis in that marine park, and then convert to hard classification. But this approach helps us a lot to make a quantification of natural hazard impact at that region and make some foundation work here because we have cyclones some months after the field work. So it was great that we tried to quantify what we lost, what we gain in such situation in coral and hard substrates, in submerged vegetation and seagrass, and sand and soft substrate. So we use beyond classic hard classification also to quantify the pre and post cyclone impact on that particular region. Big question always appear to my ears and in discussions. Can we go global for global seagrass mapping? Because people bring that we have for the coral reefs under Allen Coral Atlas, under of course several discussions on the global products, how fit to the local and regional needs, and for the mangroves under the global mangrove goals. For seagrass, to my best knowledge and experience, this can happen only at regional level due to the tides that if we have submerged or intertidal, we have to take account that. Sedimentation process, species variability. We work in the Mediterranean with Posidonia, a perennial species, but we have other very ephemeral species that you have to know very well the life cycle and the fluctuations. So to go global at once is not really advisable, rather to work local to make high quality products and assemble them to a global nice product under OBIS. Something that always is about interest to me, and I think that will trigger discussion further, is the scale. The scale of Copernicus Sentinel2 against Copernicus contributing missions like Planet Labs and Maxar, and against drone data. You can see the same meadow at different sensors, how it is visible, what you lose, what you gain. By that, you understand pretty well that depending on the needs of your mapping activities, you have to choose the best available sensor at that time based on funding, on demands, if you set the baseline for future work, or if you want to do monitoring, and by that to see how your data, your decisions will impact in the near future or far future decisions, because a very linked paper appears sorry. And here you see also again this impact about planet scope imagery in panel A against Sentinel2 composite image in panel B, that we have different aerial extent estimations for the seagrass at least, and to a certain extent for reefs, because it is the scale and the temporal resolution. And this has been quantified very recent by some good friends, Steve Kenty, who is now the mangrove EOV co-lead of the system, and Miguel Cifuentes. They map the same mangrove forest at different sensors, quantify the carbon stocks, and try to see the impact on the reported national determined contributions values. So they prove that here at the same meadow, if you use the best high resolution data you can have, you will show the real values. But when moving to coarse resolution open data, you can overestimate carbon stocks up to 1/3, which is, to my opinion and view, quite impactful in decisions about carbon credits, in the future biodiversity credits, restoration projects, and all what are under the NDC's framework. That's from me. Hope my presentation to open fruitful discussions after that.
Thank you, Dimitris. Indeed, I think you touch the important points. Scale is actually a big issue. What is the impact for these carbon accountings is a big issue. We now have Bide Rowe Davies from the University of Nantes, who will present an initial map of European intertidal seagrass. Bede is a postdoc researcher and has been working on mapping seagrass for some time, and has recently made a set of very impactful and notorious papers on this topic. So we are very excited to have you here. Thank you for joining us. Please go ahead, Bide.
Cool. I'll share my screen. Okay. So yeah, as Andrea pointed out, I'm going to be talking about our work looking at making a European intertidal seagrass map. We're calling it an initial one because, as all of the speakers have already talked about, there's quite a lot of inherent errors and issues when we're working with Sentinel data, just from the spatial scales, but also spectral scales, and then also modeling how we get that accuracy or ability to map these things with models. So yeah, this work comes out of the BICOM project, which was a European Space Agency funded project. And these are the co-authors on this paper that's currently under review at the moment. But there are many more people that have been involved with the previous work, and also some of the products that we use within our pipeline to be able to create this work. So yeah, so a little bit of background just onto intertidal seagrasses specifically, quite often overlooked, especially compared with their far more attractive subtidal cousins, but form the interface, or often in the interface between terrestrial and marine systems, therefore in quite extreme environments, going from totally exposed to totally submerged multiple times a day. And because of this, there's quite unique biodiversity and services that are provided by these ecosystems, but also are quite difficult to get to fieldwork wise. You only have a short time window where you can physically be in the intertidal area, and therefore have generally been poorly described. And when they have been described, quite often there's a spatial bias of being quite close to a normal research institute. So the idea of our work was to be able to create a method for using remote sensing that would be able to predict these intertidal habitats harmoniously, using a fairly standardized and repeatable method. And our aim is Europe. We are looking slightly further afield now, if we will, to do that. And again, repositories like invaluable for that sort of process. But originally, the process was to basically create a model that we could use to identify and classify intertidal habitats. So we created the ice creams model, which is intertidal classification of Europe, categorizing reflectance of emerged areas of marine vegetation with Sentinel2. And what this algorithm does is a neural network model that classifies vegetation in the intertidal area as well as sand and muds. And then what we then do from that is we take the seagrass class from that prediction. It has about 9 different classes. And we calculate the intertidal seagrass cover based off of work by Laura Zoffley in 2020 to convert that to a seagrass percentage cover per pixel. And then using the repository of Sentinel2, we can then go back to, well, effectively back to 2018 when data are very consistent and easy to access, and apply that at European scale, which we initially did across multiple sites. So specific sites where we knew there were seagrasses. There were seagrass beds ranging from North Africa all the way up to the Cromarty Firth in Scotland, and looked at how those sites changed over time. So initially we looked at the sort of the phenology of intertidal seagrasses, so looking at how these different sites with different latitudes were changing across the year, but then also using similar sort of analyses to look at how those seagrass beds themselves have changed with time. So how have they changed over time? And you can see the sort of the GIF going through there of Bournemouth Bay, which is near to us in Nantes. So once we've got that model and we're quite happy with it, it's got a fairly good accuracy of applying seagrass. We want to try and create some way of applying it at the European scale. But as Dimitri mentioned just now, there's a lot of changing tidal regimes. There are a lot of different factors that mean you can't just look at one image and see the whole intertidal area of Europe. So to do that, we basically wanted to first of all find areas of in the intertidal area. So we used an intertidal mask that was created by Murray et al. In 2019 as our starting point for the intertidal area. Then taking the full time series of Sentinel2 imagery, we decided to make quality mosaic. So applying the mask to those images and selecting the times where it was positive NDVI and negative NDWI, meaning that it was vegetative and or not water effectively. So using exposed imagery, then that allowed us to create composites of the whole of the intertidal area of Europe, going from the tip of Tarifa down in the south of Spain, all the way up to, I think it was 66 degrees north. It's where the Murray mask goes up to. And that we created the highest NDVI composites in that area. So selecting within the time series the pixels that had the highest NDVI and taking all of the spectral information that goes along with that, and then applying our ice creams model to that. And that created the top 5 vegetation classification maps of Europe, which then meant that we could apply our conversion from the seagrass pixels all into their seagrass cover. And what we did was we took from the 5 top NDVI composites, we took the mode classification. So if seagrass was the classification at least 3 times, we would say that was a seagrass pixel. If it wasn't, then we said it wasn't the seagrass pixel because it wasn't consistently classified as seagrass when it was at high NDVI. This obviously could introduce sort of biases, for example, with other algae covering seagrass beds, for example, like green algae, like Andrea mentioned, but also floating brown or red algae could also be an issue in that situation. But it would have to be happening quite consistently for us to not have that area as seagrass. And so this map very much, we sort of class it, and using this workflow, we class it as the maximum potential intertidal seagrass of Europe. As it looks at a time, it's taking a time window of 2017, 2018 up to 2023. So it's taking 5 years of data. But within that, we made sure that we kept hold of that information about when those pixels were taken, so that the end product at the very end, you'll see, has the date, the time of the year, and the year itself inherent within that. So from that data product, we were able to create hotspots. So looking at not necessarily seagrass meadows, because that's quite a defined thing, or not defined thing, but areas where there are high levels of seagrass within a certain area of each other. And we found that actually quite a lot of the good seagrass mapping, or what we felt was good intertidal seagrass mapping across Europe, were in these sites. So, for example, in the Wadden Sea up in North Frisia was very, very well mapped, going back quite a long time, back to, I think, Second World War imagery had been used at certain points from 1933, I think it was, in the North Frisian Sea, but then also other areas like Arcachon Bay, Ria Formosa came out as quite large areas of intertidal seagrass. And like you said, within our workflow, we maintained, or we kept, the timing of that that imagery. So that allowed us to find out at what time of the year, or at least what part of the year, we were seeing this maximum intertidal seagrass extent, and giving us another alternative view of the phenology. Previously, we did work using GAMS, looking at how things change over time, whereas here we're selecting the time that it was the maximum to look at this. And it showed a very similar pattern from what our previous work said, which again filled us with confidence, which is quite nice, that generally the further north you are in Europe, the earlier in the year, the peak of intertidal seagrass is, or at least in cover. This may not relate to shoot density or biodiversity or biomass, for example, but from our point of view, the peak is following that sort of almost linear line. This is a sort of GAM analysis using general additive models, so allows for non-linearity. Yeah, we found it is fairly linear and consistently. But then looking at the data that we had, we also wanted to look at how the proportion of the intertidal area covered by seagrass changes with latitude. And we actually found that generally at lower latitudes, you had a larger proportion of the intertidal area covered by seagrass. However, I will note that the further south you go in Europe, the smaller the intertidal area there is available. So there's a lot larger areas, extreme large areas in the Wadden Sea, for example, of intertidal area. But also that the actual percentage cover and the patterns in percentage cover changes with latitude as well, where we're seeing that actually it's far more likely to find lower density intertidal seagrasses at northern areas. I'm saying lower density, that's 50 to 60% cover from our estimates. But then when you go further south, the seagrass beds themselves are tending towards being higher density within pixel of Sentinel2. So that data is, like I say, and these analyses are under review at the moment with remote sensing environment. But our end goal is, as Lena's been really advocating for, and I think most researchers are quite keen for, is having these data products not only in research papers, but open access. So what we've decided to do is actually create a Google Earth Engine product so that end users can inspect and interrogate this data themselves. And like I said earlier, it gives the seagrass percentage cover. It also gives our uncertainty estimate from that, what day of the year it was, and what year it was for those things. And you can, in the app, you can actually click and see a single pixel and see what day of the year it was and things like that. But then going forward, we're sort of thinking that this can then, once we've got this product there, we can actually identify some gaps in it. We know it's not perfect. We know it's an initial map, and we're just keen to, once it's out there in the open for people to inspect it and say which bits are wrong, which bits are right, and then improve the workflow itself so that we can move forward to maybe more global estimates. But yeah, that's all for me. Thank you very much.
Thank you. That was quite impressive work. I'm sure many people are interested in looking at this data that you have and might make available. So now we will have Mar Rocamora from ICMAN CSIC will present beachcast macroalgae mapping using Sentinel2 and future perspectives. Mar is working on her PhD and has made several notable works, including one that I think she will focus on, on the problem of beachcasts, which is when algae, seagrass or macroalgae are washed ashore. Mar, the floor is yours. Thank you.
Thank you very much. So yeah, we're going back to macroalgae. Yeah, you're right. I'm now focusing on the work of Rugulaptrix Okamurae It's something that we did a pilot study 2 years ago, almost 3 years ago, and now we're trying to scale up this solution. So for what? Mainly to contribute and enhance the beach guest management, because they're generating huge biomass on the beach. So this is the species we're focusing on, is Regulopteryx ocamurae. It's an invasive alien species that arrived to the Strait of Gibraltar 10 years ago and is being expanding both to the Atlantic and the Mediterranean Sea. As you can see here, this is from last year, so there are many other places that are invaded right now, but you can see that it already jumped into Italy, so it's on the way. This is the quantity of biomass that generates just in a day, and the municipality has the competence to remove it and clean the beach if they want to keep tourism in these beaches. And this is the potential expansion model that was developed 4 or 5 years ago. So as you can see, the Mediterranean is just a matter of time, because it's expanding towards that part, and also the African coastline and the African West. So I'm going to be focusing first of all in this case study that we developed, that we were integrating multispectral sensors and hyperspectral measurements to try to quantify this beach cast, the dry macroalgae that was outside the intertidal parts. We were trying also to account the floating and the subtidal until 5 meters depth. So for that, we were trying to assess the accuracies of the satellites for macroalgal detection in these 3 stages. So what we saw was different spectra in wet and dry algae that determines 2 different workflows. For this algae, we found a peak in the 710 nanometers, and 2 different behaviors on floating and wet and the dry algae. So it is really important to separate the workflow on the intertidal dry and then the floating and subtidal part. As you can see here, this is the beach cast on the intertidal and dry beach, and this green line is the spectral radiometer. Here the yellow part is Sentinel2, and as you can see here, these 3 dots here are the reddish bands that are really important to detect this beach cast and are not existent in the Landsat imagery. So this is a really big advantage on Sentinel2. And we also compared the accuracies of mapping in these 3 platforms, and we saw that 80% of the total accuracy was obtained with Sentinel2, so we can really detect and quantify the area of this huge biomass doing this type of methodologies. But yeah, we have to really separate the methodologies into different workflows. So this was a case study, a pilot that we wanted to explore first, but how can we create an operational macroalgae beach cast monitoring platform to detect, track, and assess this macroalgae in an operational way. I'm going to go through these 6 points. First of all, repositories. We've been talking about OBIS a few minutes ago, but currently there is not a dedicated benchmark dataset specifically for training beach cast macroalgae algorithm models using Sentinel2 data. So this is the most important part. We need a spectral library of these coastal habitats to success in these scalable solutions. So available data right now, well, ice creams is being now showed by BID. That was from last year, and it's a really nice data set that we're trying to implement as well in this case. I found the coastal ecosystem spectral library, but it's very limited. It's only focusing the east part of America and part of Portugal. For GBIF, OBIS, and ASIN, that in this case is the European platform for invasive alien species, I found that was like a lot of duplicity between the platforms, that we have to create data. And it's kind of difficult to use it for Earth observation applications, because we really need a lot of accuracy in the date and time of the observations. There has been citizen science projects developed in the south of Portugal as well with this algae that is working quite fine because we can get absence present points from there in the moment. But mostly we have to rely most of the times in photo interpretation. We have to create these spectral libraries in its case. So there's a gap of information on this that we have to assess, collecting and unifying all this in situ radiometry and pairing it with Sentinel2 data. Regarding the remote sensing approaches, in this case, I would go for a single image approach. So we take one image, we filter and do all the processing. So we have one image per product, but depending on the computational effort and the workflow that we are following and the scale and the variability of these bit cuts, we can also go for multi-temporal stack of months or seasons. So we can see like accumulated impacts on some places. So first of all, we need to delimit the area. I've seen a lot of works that use geometries itself, like manual geometries. But if we want to scale this solution, I think we should go for something more automatic. So this is an approach that can be one of them. So taking the coastline of like a global coastline, we can buffer it depending on if there's a tidal place or not. And then we can use the NDWI to remove the water. That's the easy part. So we can account as well for tide variations depending on the tide level. And then for the terrestrial parts, we can use Sentinel-1 data using the 19th percentile of the horizontal polarization. And then we can do like this type of mask to take the dry bits and the intertidal. Why not using the scene classification layer that is already in Sentinel level 2A? Because this is what we got previously, but this is the classification layer that it's available by default. So it's not really useful for the case. For the image processing, I think it's crucial to have a unique pipeline in one platform, not jumping from one platform to another. And for the bands, 20 meters bands, we will downscale it to 10, but band 1, for example, that has 60 meters, we will just not put it in the model. So this is an example of how can we develop a model using these bands, let's say, the NDVI, the WI. A lot of people also uses the maximum colorful index that accounts from band 4, 5, and 6 using this red edge box. And the normalized different chlorophyll index also works quite nice. Then after the per pixel cloud filtering, we can have one data set, just binary, as we were talking before. So we extract the pixels, we train the model in binary, non-bitscast and bitscast. And then with a second classification, we can specify which type of bitscast are in these pixels selected in the first model. And then with an independent data set, of course, in situ, drone imagery, whatever, we validate, we do the accuracy and uncertainty assessment. So variables of interest that we can get, accumulated impacts along the coast, area quantification, percentage cover that we talked before, spatial uncertainty maps, and for the third dimension, the biomass, because here, as you can see in the picture, these bitscast can be really, really thick. We can also try using Sentinel-1 to take the difference. And with that biomass quantification, then extrapolate to carbon if we have carbon in situ measurements. And to scale the solution, I am using Google Earth Engine in this workflow, but we can use other cloud computing platforms like the DS of the European Space Agency. Use a unique pipeline. For this case, we can use level 2A imagery, so we are avoiding the computational effort of the atmospheric correction. And then, as did also showed, like creating these apps, so for decision makers, it's really available, this information to be used. So last, challenges and opportunities that I think we have, this benchmark data set to really measure model efficiency, accuracy that we're developing, plus this data of cover biomass carbon. In terms of resolution for this algae, I think it's enough resolution, Sentinel2, for what we are studying, but other patches will not be accounted. But I think this huge biomass is the one that needs to be managed most of the time. So we don't really care about small patches. And to maximize the potential, of course, this global scale monitoring using cloud computed platforms for intertidal level 2 and for subtidal and floating level 1. And just to make just like a call, this algae is also gaining some terrain to Posidonia oceanica seagrass meadows in the Mediterranean. So it would be nice to start like taking information, in situ information, to assess in the future the impacts on the regression of these seagrass due to the macroalgae. And that's all for me. I'm open for the further discussion.
Thank you, Mark. Yeah, it's I think a very interesting presentation. It's a quite impactful phenomenon that it's taking place in because the beaches are full of, and tourists are the first to complain. So thank you very much.
Thank you.
So now finally we have our final talk. We have Chuan-Min Hu from University of South Florida, who will present Sargassum Watch from Space. Chuan-Min Hu is a renowned researcher and well known for many of you, with many years using satellite to study algal blooms, oil spills, coastal and inland water quality, and global change. He's going to focus here on the detection of floating sargassum. Chuan-Min, please. Thank you for joining us. I know it's very early there. Go ahead.
Do you hear me? Okay,
Yes,
great.
you can.
Thank you, everyone, and good morning, good afternoon. So as Andrew said, I'm going to present or introduce our Sargassum Watch from Space system, based on satellites. I acknowledge the contribution of my co-author, Dr. Brian Barnes, and many other co-authors and collaborators. And this has been a funded of multiple funded projects, by multiple agencies. We have spent more than 10 years of effort on this. So it's quite mature, but there is still room for improvement. So I notice most of you have talked about benthic macroalgae, seagrass, and intertidal macroalgae. This is different. This is the floating macroalgae. So sargassum is one type of a floating macroalgae. So on the good side, it is a critical habitat for many marine animals. But on the bad side, too much of a good thing on beaches, in coastal waters, can make it very bad, for environmental pollution and many other problems. Now, how do we monitor this and track this from satellites? So here are the principles. I show this for sargassum, but it applies to any type of floating macroalgae. So basically, you do in situ experiment to determine the percent cover or biomass density, and at the same time, you measure the reflectance. So there's a one-to-one relationship between percent cover and reflectance. The spectral shape is the same, but the magnitude is different. So we establish a relationship between percent cover and reflectance and apply that to satellite data. Okay, so this is the principle. And here's the framework. There are different technical steps to map macroalgae or sargassum. And first, we want to detect anomaly, and you visualize the image. Do you see anything that is abnormal from the background? That's a visual detection. Then, what is that visual anomaly? Is it caused by seagrass, floating seagrass, or floating macroalgae like a sargassum, brown or red algae? Okay, that's a spectral discrimination. And then we apply a spectral unmixing to determine the percent cover within a pixel. Okay, so that's on the native resolution. But finally, we want to remove gaps due to clouds, due to sun glint, many other factors. So we put a lot of images together to have a composite. Okay, so that's the framework, and I have an illustration to show how each step or how each type of a data product looks like. So the first is visualization. You can compose the NDVI or floating algae index or any type of index image. You can see this image features. Okay. Okay. Then you can detect or extract the image features using an algorithm. And you can use a threshold-based algorithm or use machine learning or deep learning, AI, whatever. So you extract these pixels and paint them in red. So all these red pixels are sargassum-containing pixels. Okay, but it's just the presence absence. Okay, without telling how much. So next step is we unmix every pixel to tell the percent cover within each pixel from 0% to 2% or more than 2% if you can interpret the color code. Now we put many images together to have a percent cover for each grid. Okay, so to remove that gap. So those are the basic concepts, framework, and steps, data product levels. So now, how do we tell what that is? Is that sargassum, some type of floating matters like a pollen aggregation or trichodesmin scum? We look at the spectral shape. Okay. The spectral shape of each different floating macroalgae or floating algae scums is different. So we compare the spectral shape with a known library to tell, oh, that is the sargassum, not something else. And the magnitude or the spectra tells how much. The spectral shape tells what that is. The magnitude tells how much. Then these are the principles. So back to satellites, we have a lot of operational satellites operated by different agencies. We download all those satellites data, okay, in almost near real time with a few hours latency. We process this data and make products online to serve the people. Okay, and so here is one type of product. It's daily or 7-day running mean sargassum macroalgae percent cover. So we produce this type of images every day in near real time online, and you can focus on individual region and click on the region and do your own animation and bring to your own data layer and to continue your analysis. Okay, so this is 1 year of animation. We also, based on this system, we also produce monthly sargassum outlook bulletins. Every end of the month, we summarize what's going on in the past month and what's likely going to happen in the next couple of months and distribute to users, subscribed users. Okay? And we also produce statistics of the current month compared to his history using whisker plots, using stuff. So how does the current month in brown color compare with history? So people have a direct feeling of, oh, this year is a major year or is a moderate year. Okay? But there are data gaps from that system because the sargassum is really patchy. Okay, in the ocean, it rarely covers a full pixel. Okay, for medium resolution satellites, it's less than 1%. Okay, most time it's less than 1%. For Sentinel2, it's a couple of percent to 10%, rarely a full pixel. So we develop Sentinel2 algorithm, apply the same principle to Sentinel2 data. So here is MODIS. You see the large pixel in Florida Keys. And so there's also data gap. Now with the Sentinel2, we can fill that data gap. We can see better. We can see smaller pixels. Or these are already aggregated to 1 kilometer. Otherwise, you would not be able to see this tiny sargassum slicks. Okay. So then we apply the Sentinel2 algorithm to a real-time data production. Here is our website. So every day we download the Sentinel2 data and produce and distribute data products online in real time in just a few hours latency. Unlike benthic macroalgae or seagrass, floating algae moves fast. It's very dynamic. If you don't capture that today, perhaps you will lose that tomorrow. So we have to produce a daily image to capture the dynamics. So here is the sargassum map for Florida Keys. If you click on the image, you see the tiny little image slicks, swirling, elongated images, tiny slick. Those are sargassum slicks. So people have more detailed view every day. Okay. So here's the basic summary. So in the open ocean, sargassum is a critical habitat. So we monitor this open ocean sargassum using medium resolution satellites like MODIS, OCHI, VIRS, OCI. This is the PACE OCI. In coastal waters, sargassum turns into a harmful algae. We use high resolution sensors, including Sentinel2 and SuperDove, to zoom in the coastal area. So this is just one example of how we monitor and track floating algae, the Atlantic sargassum. But globally, there are many types of floating macroalgae and floating microalgae scums. The principles are the same. They're floating on the water. We unmix the pixel, we tell what that is. So once there's a need, we can just expand the system to have a real-time monitoring system for a certain area. With that, I'll stop. Thank you.
Thank you, Chuanmin. Yes. Again, another great application of satellite data. In this time, a bit of a different matrix, but equally important. I think now we can move to the question and answers. We already have a few. Please, everyone in the chat, try to move your questions to the question and answer box that you have here in the bottom of your screen, and we can follow better your questions. I'm going to start here at the beginning. I think this one is from you to you, Lina. Apart from the drones and spectrophotometry, are there any other applications of electronics and automation in your bioequation project? I believe there are. We have many studies ongoing, so don't know if you want to reply this. You have.
Yeah, I was just going to say, I think you are the best person to respond to this, at least if the question is related to remote sensing.
Yeah, I mean, we do have other applications. We are aiming to use remote sensing in other living labs, including Tuscan living labs and also in the Baltic Sea. So you can always contact with us and make your question. We can try to answer, to explain better what we are doing. There are, I am aware that other groups in bioequation are also using electronics and automation for the project, like video cameras, something like that, and even acoustics. So please get in contact with us. We can share what we are doing.
Yeah, and I mean, we're also very much trying the whole data pipeline with the essential ocean variables for both macroalgae and seagrass.
Yep. So now we have Balogun to Elizabeth. Under your presentation on future works, you talk about fit-for-use datasets. What standardization is being proposed to ensure that field researchers obtain fit-for-use datasets across multiple platforms? So I think this is about data being operationalized between platforms, between OBIS and others.
Between OBIS and, sorry, what was?
Other platforms, like I imagine there are other repositories.
Mm-hmm. Yeah, so there's OBIS and GBIF. We already use the same data standard. It's called Darwin Core. And then if you're talking about specifically what I mentioned, of tagging data sets as fit for use. So first of all the data in Opus is already standardized across multiple data sets. And this future work that we're working on of labeling data sets that they're useful for a particular purpose. This is a very early work in progress for us. So I don't know if I can answer that part of like what exactly are going to be the protocols for how that's going to work. But I know there's some work within the data standard community with specific like having categories for data sets. I'm not sure if that answers the question, and perhaps there could be like some clarification of what specifically you meant by across multiple platforms, if that's indeed across
Yeah.
these other multiple platforms or.
Okay, Valerian, if you can make it clear, maybe we can return to this question after. Okay, this one is a big one, and I leave to one of you speakers to decide what is the capability of macroalgal mapping methodology in separating macroalgae from seagrass. Who would like to take this one? Dimitris, Bid. This is an important one, and I guess this is one of the major limitations in doing some kind of operationalization
Can
at
you
a
repeat?
global level.
Sorry,
So
Andre, can you repeat?
this is about methodologies in separating macroalgae and seagrass.
Okay.
So
Based on my experience, that it comes from different waters from other regions, my macroalgae are not green or almost not green. Or where my macroalgae grow, we don't have seagrass and we have Posidonia, most Orchimodosea. So I have managed to separate brown macroalgae from seagrass. However, this is not the case in Atlantic. Bede has very good experience on that, and Mara as well. So it is region specific the problem, and spectral specific. What means that we use multispectral sensors, not hyperspectral sensors, and the available hyperspectral sensors come at a cost of resolution. 30-meter pixel size doesn't facilitate the separation. So we are in a period that we expect high resolution superspectral sensors, but are not available now. So you need to have high quality field data for all your classes and to try to understand how spectrally are separable. If not, then you cannot.
yes, I agree. Yeah.
I want to add something to what Dimitri said. I totally agree. This is a regional problem. If you apply a technology or algorithm blindly without knowing your area, very likely you have error. You have to know your region. What is the possibility? Worldwide, there are many types of macroalgae, different types of seagrass, right? Different colors. So, but in a specific region, perhaps you are restricted to only either this or that. That makes the problem simpler. But even though a lot of times, this macroalgae or seagrass have only a few percent of a cover within a pixel. So even if you can unmix or discriminate a pixel, say at 50% or 70% cover, down to a couple of percent, can you still do that? Maybe not. But you can do just the intense pixels, focus on those large pixels. Then with some educated inference, say if some pixels in this area are macroalgae, then the rest of smaller pixels are same type. So it's a combination of regional knowledge, of educated speculation. Applying a technology blindly is pretty risky. That's what I want to say.
Yeah. So in situ data and local knowledge, it's a must. Did-
Yeah, because
yes.
I was just going to follow on from what Chao Min just said. I think it's really, really important to not just apply technology blindly, like he said. It's one of the things that we sort of came up across in our project was actually looking at, what algorithms can separate. Specifically for us, it was green algae and seagrass. But actually, in the process of developing the algorithms and stuff, we found that actually our better accuracy came from having data from a wider range in our training data. So I'm very much talking in intertidal, but also specifically, from the modeling point of view here, that when you have an awareness of what could occur in the range where you want to apply the model, you sort of have to have training data that would represent that effectively. And one of the things we've seen in a few papers recently is, this idea that in a certain region where that it's only one type of algae, you can apply NDVI or some other index, and it's relatively blind. But because you know the regional information about that bay, you can infer that it is seagrass or macroalgae or whatever you think it is in that bay. However, then when you scale it, that's when you run into issues. So I think the idea of scalability needs to come. Well, it needs to be aware of what's diversity in your system there is, and also account for that as you're going forward.
Yep. Thank you. Dimitri?
Yes, I will touch upon what the previous speaker says, that fieldwork is the most important part. Go out, see how your system behaves, see how the light travel, subtitle or intertidal, understand what you have, and then just be very clear about what you are trying to do with understanding also the sensors side of you, what the sensors you are trying to use can offer to you.
Yes, coming back to the basics, right?
Exactly.
Go in situ, which takes time and money, and but it's yes, it's
And to top up here, as Mar says, she tried to find data from all repositories, and if the data are here, we say we have a lot of data, then not all data fit for that purpose, because the metadata are not curated well. The accuracy of the GPS is not documented. And people doesn't know even here maybe that even the satellites from pass to pass, you have errors, geolocation errors, that technically you need to correct them to do proper change detection analysis. Otherwise, you will introduce errors inside of half pixel in unknown direction sometimes, and then you end up with positive or negative findings that cannot be used by policymakers, but technically might be errors from the geolocation and not real changes. So there's not a lot of need of understanding the sensors technology, the geolocation, the fieldwork. That's why remote sensing is not just only machine learning and coding. You need also to be proactive on ecology, physiology, lights, and fieldwork.
Thank you, Dimitris. Okay, I'm going to come here. There are a few questions for me. Andrea, which product have you used to create regression model? Well, the product of Sentinel was the Sentinel level 2A data, and the regression model, we tested several, linear and nonlinear, and we came with the quadratic, which was the one that just seemed to fit better. I have a question here about UAV and the battery. Unfortunately, it's not me that operated the UAV, but you can always reach out to me and I can try to find this. Do you have some field tips here for sampling with a UAV to cover large areas as much as possible? I mean, maybe Dimitris, I know you work a lot with drones, so maybe do you have some field tips here for sampling large areas with UAV?
Large areas with UAV. I have experience from quadcopter drones from DJI and some customs. There are two sides of the science here. One is how much money you have to buy fixed wing drones to have a long range. The other is about licensing and regulations. Because to fly drone is not just simple, you go and buy something and fly it. You have to fall into regulations of local authorities, safety about the airspace. So sometimes the investment that you will do to buy long-range drones to cover big areas might be quite costly in comparison to rent an airplane and with the same money to cover 10 times more area. This is my experience from hyperspectral drone because I had the luxury to use one that cost 500,000. It fly 9 minutes. It give you some square meters of data, and with the same money, I can rent from a company an airplane and fly 1,000 square kilometers of hyperspectral data in LiDAR. So it is not always a drone the best way to cover large areas.
Right. Thank you. Okay, now I have here something about to me about comparing my classification results with previous studies. Well, that's a work in progress, and we will try as much as possible to compare with others. I think comparing models and classification models is also a topic of interest that will come up in the near future, I guess, because with models coming up, I think at some time there will be some type of intercomparison also between models, different classification models. So now here, how and where can your product be accessed for experimental use in the Baltic Sea?
So generally, the actual product itself is not available yet, but we're planning to have it, I think, either on Zenodo or any other data repository platform. The case about the Baltic Sea is that there's almost no intertidal area in the Baltic Sea generally, if you compare it to the rest of the Atlantic coastline. Even in the paper, we have the Mediterranean included, but again, we don't talk about it because it's magnitudes of difference. So from the Baltic Sea, I think it depends where you define the Baltic Sea as being. I think we may not actually have it included in our models at all because we sort of stopped at about where Copenhagen is when you go up and over Denmark. So yeah, Baltic Sea might not be available. The model itself, Ice Creams, is on a GitHub repository, open access. You can retrain the model with the training data that we've got. It's all on a GitHub repository on my website or just on my GitHub that has the training data itself, the code to train the model, and then also the code to apply the model to an individual Sentinel2 image, as long as you've got an intertidal mask yourself. What else was I going to say? I think that answers the question, I think. Yeah.
Yeah, thanks. Okay, this one about distinguished macroalgae and seagrass you already discussed. And then we have the Elizabeth, are you answering this? Okay, so it will be covered online. By written, we reach the end of the questions. So now we can move forward to our discussion between us. We have about 20 minutes. So I'm going to launch one question for each of you, the same question for each of you. You have about 2 minutes just to decide how we want to do. So from your perspective, what is the single most important thing we need to do to improve or solve to fully take advantage of Sentinel2 or remote sensing in general, the open access remote sensing, let's say. Who wants to take the Mar?
Yeah, I would say it's about in situ data again, like having this benchmark data set so we can test, model, and compare the different methodologies we're using. Because data is really like dispersed in a lot of platforms, and maybe like focusing in Europe, having like an European database in that case of macroalgae and seagrasses, we can compare the accuracies of our models and so on. And about scalability, for me, using cloud computing would solve that part. But I think now the limit is mostly with the in situ data set.
Good. Dimitris, we're going to
Yes.
speak. So.
Well curated in situ data associated with spectral information, very well documented metadata, and to cover as much as possible potential classes that can be distinguished by remote sensing now, but also having in mind the future. So to collect ones used for many approaches is very important when we need to go out and collect data and make them open through proper databases, well documented, because for me it's very important. When I receive data, I'm looking for documentation, which mostly is lacking. So for me, are no data at all.
Can you, because you are already two saying the same, more or less the same thing, can you elaborate more on what kind of data, ideally, what ideally would be your curated dataset type?
Ideally, a data set about seagrass occurrence in points and polygons, and also absence of data, because some models that are available require high quality absence data, and nobody is recording the absence of the target. Everybody's looking for the presence, but the absence doesn't care. But we have seen that the absence matter a lot in modeling development as well.
Thanks. So, Lina, what?
Yeah. Did you say Lina?
I said, just I was
It's
trying to remind you the question, but please
yeah.
go.
And I agree very much with both Mar and Dimitris. I mean, when it comes to this high quality in situ data, it's so important. And when it comes to fair data, definable, interoperable, accessible, and reusable data, it is actually quite difficult compared to, or it's not difficult, but it's quite a lot more than we usually do with the data. So, I mean, to really record that metadata, for example, your GPS accuracy, maybe even what GPS you've been using, the method you've been using, and all of these things, and not just making the data openly available. It doesn't mean it's fair. It really needs a couple of extra steps. And OBIS, for example, they have a lot of support also of how they can help us to actually make data fair. And with more data available, the better products that we can do. And I want to also echo what Dimitri said, that, I mean, absence data is also very, very, very important.
Okay, before I'm going to Chuanmin, because I know he needs to leave, I think 10 minutes before. Chuanmin, what would be your single most important thing we need to improve or solve to fully take advantage of remote sensing Sentinel2?
Well, there are two things, on the research side, as a researcher, I totally agree with what's just been said. We need high quality in situ data, a library. I emphasize high quality because not every data point is equal, can be really used. But on the, I want to talk about another side, on the user side. They really don't care about how we do this, right? They just want the data products, the quality control, the data products to be accessible in a user-friendly way so they can make their own use. So I want to emphasize on this side. I see several questions being posted in questionnaire, where do I get the data? Where do I get the data? Yeah, so that's in line with what I just said.
Thank you. Elizabeth, from your side, which you are the one that receives the data, what is the most important thing you consider?
Yeah, kind of like to echo what everyone else has been saying is to have complete metadata, as much information as possible. So information on the exact methods that were used. If you use the best practice, like include the links to the methods in your data set. Like there are data columns that can be used to point to additional resources. So like I mentioned, like associated media, making sure the data set that's going to OBIS has as much information as possible. Yeah, so the methods, what devices were used, the uncertainty, and all this can be really useful to create high quality products that the user will need.
Right. Viv,
Yeah.
just missing you. I think so far most of everyone said in situ data. If I got the option to reply, I would also say it's one of the most important things. What is your thought?
So yeah, I totally agree. It's in situ data, but I'll try and talk about something else because everyone else has focused quite nicely on the actual data itself, but also sort of focusing in terms of methodologies and best practices, like Elizabeth just said, of following best practices, being able to cite that. But one part of that is also propagating the uncertainties that we have when we've got these multi-step pipelines that are then creating an end product for users. Because this is one of the things that I see really regularly is a seagrass map that's, whether it's a habitat suitability model or a random forest model on Sentinel2 imagery, you have the pixels that are classed as seagrass and the pixels that aren't. But both of those methodologies, whether it's prediction or description, have ways of giving some form of uncertainty or form of probability within those models. They're not perfect, and I think that needs some development from a research point of view. But I think it's also really useful for the user and end user to be able to have that information as well. So not just the classification of the habitat itself, but saying how confident are we? And when we're not confident, put our hands up and say, yeah, from this imagery, we can't actually say whether it's seagrass, macroalgae, or whatever, whether it's to do with sunglint, whether it's to do with cloud or whatever. I think that's a really important thing to be propagating within any of these, especially global products. We're saying, I used the intertidal marsh from Murray et al., and they had an accuracy of 82% accuracy. Very good, but when you go to the regional scale, you find places where it's really bad and some places where it's really good. But being able to say what their certainty was in those regions is also really important, especially for end users who are probably working at the regional scale, not at the global scale.
Right. So now that we all kind of agree on also data, in situ data being important, how now this is for whoever of you wants to take this one. I mean, everyone wants data, but taking data, it's very difficult. And in the owners of the data, they want to have some property. So it's very difficult for this to be shared. And I mean, chlorophyll, it's the standard biological variable that it's mostly available, but then macroalgae and seagrass, maybe not. So where are we in the availability of these benchmark data sets of locations, occurrence, reflectance of macroalgal seagrass, and how can we improve that this becomes more available in the future? Any strategy to convince the owners of the data to publish? Okay, so this was a difficult question, and none of you. Okay, Lina, yeah.
It's okay. I mean, it's either me or Elizabeth for this one. But for example, now when you upload the data to OBIS, you will get a digital object identifier, a DOI. So it is possible to get acknowledged for the data that you upload. And I think also that we need to communicate more with funders, for example, that this kind of data, I mean, this in situ data is very, very valuable. Sometimes even more valuable than an output or a scientific paper, because this is data that can be used for all into the future, which is amazing, and by a lot of people. And we spend so much money in collecting data. So we should really celebrate every single person that dare to share their data.
Yes. Thank you, Lina. Okay. Let's go for another question.
I think Bede also had his hand
Yeah,
up.
sorry, I had like a
Okay,
bit of
sorry.
an add-on to Lena there. I have quite strong feelings on this, and I think there's also like a slightly ethical decision to be made, considering the fact that the vast majority, not all, but the vast majority of research that goes on, especially in Europe, is from public funds. I think then the outputs have to be public and they have to be in that realm. Otherwise, it's just unethical from my point of view. And again, that goes back to methods, that we're applying the most appropriate methods and all that sort of thing. But the fact it's open at the end and accessible and reusable and all those sorts of things, that's an ethical decision as well that I think needs to be made.
Yes, I will agree with Bit that public money, public data first, then the credibility of the data sets is still in a gray zone. I have experience from my data sets, from my PhD, that I upload them in SBIF. This entered to OBIS. Even the paper that has the data has only 20 citations now, 10 years. The same data set has been downloaded almost 2,000 times, which is way further the value of the dataset from the paper, because people go to SBIF, look for biodiversity data, take everything, and this everything has been 2,000 times. So it's interesting how we can also work with OBIS to take citations, if we can name that, from datasets, or the funders, as others says here, or in the proposals to show the value of open data through these portals, and not ask for gold open access publications that cost 3,000 to 10,000 euros.
Yeah, so we also should maybe make the life easier, or try to do our best to have this data well organized and also easily accessible for everyone taking data. And also then the platforms also need to be friendly and to allow easy access to this data and findable. So now I'm going to move to the final questions, which is about stakeholders. So, okay, we agree that we can do something at the level, at the local scale. So we agree that we understanding the region, and we can develop some products that are available for stakeholders. But how can we pass this message for the stakeholders, for them to use? What is your how do you see the uptake of satellite-derived products by stakeholders? And what has been your challenges in reaching the high level where you want to say, we found changes, we want to report these changes? And does any of you have an opinion on this?
Yeah, in my case, in this invasive macroalgae, I think it's something, well, in all cases, that has to start from the beginning. First thing I did was asking them, like the public institutions and the regional institution and the ministry, like which type of information they had and which type of information they needed. So we can design the workflow according to that. In this case, they needed, because when they see that they can get money, then they're more interested on that. In the beginning, no one cared about that. And now, they have the opportunity, the municipalities, to, once quantified that macroalgae, ask for the impact, the accumulated impact of that algae, for money to the ministry. So they can be, somehow compensated with the damages. So yeah, I think it's a matter of getting in touch with them and then check first which information that they have, so we don't have to double efforts in that sense.
But it's a big effort, right? One side
Yeah.
you have to do the research, and the other side you have to be knocking on the doors to get this data used. So how can we make this life easier? And I mean, because I have here you speakers, I think all of you have almost products that can be used for some stakeholders. Have you how it has been your interaction? For example, you with I mean, these maps that you are now producing for Europe, I guess they are taking a lot of interest, no? Have you been in contact with stakeholders at users for this?
Yeah, small amounts mostly because we've talked especially with some of the, like I say, in the Wadden Sea, there's the regional government and municipalities that do a lot of sampling there. They're actually involved in the paper itself that's under review. But previous work, we've also had quite a lot of stakeholder engagement specifically to do with the seagrass bed in Bournemouth Bay. There was a local group that were they were counting Brent goose occurrences, and actually we noticed that the Brent goose, because they're feeding on the intertidal seagrass, they were very highly linked. So they were using the data products from that. But from that point of view, it went very well because they were involved from quite an early stage in the project. And I feel it's quite difficult to get to a stakeholder and say, look, here's a map that we've produced, you just have to trust it. Whereas actually if stakeholders may be involved with the process or has given some input either in training data or as validation steps, like a lot of the validation from ice creams comes from it's other researchers and stakeholders that aren't in our specific research group that we reached out to and said, we've got this product, we think it's quite good. Could you provide us with data, or just even if it's not quantitative but qualitative assessments of these different maps? And I think that then gives a little bit more trust, I suppose, into the products if the stakeholders themselves are involved in somewhat in the process. But again, that's very difficult when if you're trying to do a global or continental map to say who's a stakeholder at the continental level, then going up to quite high levels. But yeah.
Okay, good. Thank you. We are reaching the end of our workshop, so I just want to ask you a final message on you about this subject. What would you have as a final message regarding the way forward and the possibilities of using the remote sensing for macroalgae and seagrass monitoring? Dimitris, please.
Understand the limitations.
Okay.
Not overselling and don't self-fume. You have to be very precise to understand what the limits of the technology we have. And to co-work for ideal sensors for coastal habitat mapping, which is a dream, but when I talk to high-level ESA persons, I already say, okay, but it's a very narrow part of the world, so there's a need to convince them about the need of sensors that are good for coastal systems.
Thank you. You can also please send the message of, what are the new technologies that might come on handy in the near future that you think it might change the way we are now progressing? You mentioned about high resolution hyperspectrometry.
Yes.
That would be a big advance, I think, for everyone. Anything else?
Commercially, it comes available slowly, not at the hyperspectral spectrum, but in the superspectral, so 25, 30 bands. Full hyperspectral spectrum at high resolution. I know that at the military domain exists, but not in the commercial sector yet, or in the open sector yet. And what is foreseen from NASA and ESA as open data will be at 30 meters, not high resolution. So for me, it's a matter of allowing the military technology to come to civilian technology. Just look back to the GPS.
Thank you.
Nothing else. I know more, but just look back to the GPS.
Thank you, Dimitris. I'm going here by my order. Bid, one last message. Do you want to share any thoughts?
No, I think we've covered it all. Really, really showing your methods properly whenever you're creating these data products is a real pet peeve of mine. That you find what looks like a really good data product, but again, if you don't know what the method was to collect it, and the accuracy of, for example, the GPS is, if the default GPS thing is 3 meters or 6 meters, or in other websites the default is slightly different, but yeah, having that uncertainty in measurement is important, and methods of how you got there.
Thank you, Bill.
And here, just to add to the GPS side bit and the team here, put the model of the GPS for the future. Put the model, GPS, Garmin, whatever. So in the future, you will Google to find specifications. So it's worth, I think, that.
Thanks. Elizabeth, one final word from you?
Yeah, not too much else to add, but yeah, just reinforcing having like complete information on the metadata and everything involved with the data set, and making sure that gets into open access global repositories.
Okay, before going to Mar, just want to ask everyone to, if on the audience, if you can fill that survey that we put on the chat. You have one more week, so please go ahead. We will be greatly appreciated. So, Mar, please give us any final thoughts.
Yeah, more about what we already said about sharing the information, checking that quality of the data, flagging it, and then yeah, creating this benchmark data set that I've been saying all this time, to blend it to Sentinel2 applications and scale it.
Thanks. Lina, you're the last.
Yeah. If you see anyone share a good data set, celebrate them in social media and tell them how amazing they are. So we start getting this positive feedback for people doing the right thing. And for the way forward, for the coming 3 years, I will work super hard together with Elizabeth to create a lot of different types of training for how to make your data go into these global repositories to actually create fair data. So that is a promise from our side.
Wow, that's perfect. Thank you. So we reached the end of our session. Thank you very much for all of you for your active participation and questions, and a special thanks to our amazing speakers for sharing their work and insights. So thank you very much, and hope we see each other in the future and we have great results in our work and continue progress forward. Thank you.
Thank you very much.
Thanks very
Thank you.
much.
Bye-bye.
Thank
Ciao.
you.
Thank you so
Bye.
much, everyone. Thank you, Andre, for a nice arrangement.
Yeah.
Mhm.
Okay, bye-bye.